Hello friends, this is Dr. Gajendra Singh Pachlore, Assistant Professor at Department of Zoology at Vinayak Vigyan Mahavidyalaya. Uh, Today we are going to see at the topic cleavage. So uh, let us move ahead. What are the objectives of our today's topic? This topic is basically designed for the students who are studying zoology at undergraduate and postgraduate level. This topic basically covers the first phase or initial phase of a developmental biology, which of almost all the living organism, which basically covers of the first initial stage, and it covers in detail the cleavage patterns and its significance and importance. We'll move into the next one. What are the learning outcome? After the completion of this topic, what we are going to learn out of this is what we are going to understand is the definition of the cleavage, what exactly the cleavage means and how the cleavage begins. You're also going to understand the different patterns of cleavage. Pattern includes the holoblastic, that is complete cleavage, and incomplete cleavage, and different types of cleavage that has been found in different sorts of organism, ranging from invertebrates to the vertebrates. At the end, you will also able to understand the significance and importance of the cleavage. Now, let us come on to the first definition of the cleavage. What does it suggest? Cleavage basically suggests that it is basically a kind of a division. It's a cellular division which is little different from the division that we usually see, that is meiotic division. This cleavage is having a mitotic division, or the cells are going to divide mitotically. That means the number of chromosome will remain the same. When is the cleavage going to occur? The cleavage is going to occur after the fertilization. What does that mean after fertilization? That means fertilization, we need to understand first of all, that is the fusion of egg and sperm. This is usually termed as a fertilization. After fertilization, the product that we are going to get is a zygote. This zygote is a diploid cell. It will be having a 2n number of chromosomal composition. As egg was haploid and sperm was also haploid, their fusion will result in a diploid zygote. So basically, this single cell that we can see here is actually a zygote, which consists of a nutritive tissue. We can call it as a yolk. Now this yolk is very important. I usually call it as a tiffin box. As a tiffin box for a long journey, whenever you are out, you basically carry along with you a tiffin box that is going to nourish you in the long journey. So for an organism, the movement or the transformation of this organism from zygote to a multicellular organism is just a kind of a journey where it will be requiring a nutrition or a food. So from where it is going to get the food, it is going to get the food from this yolk. Now, uh, having been said that, you are you also observe that some of the organism develop inside the mother's womb or inside the body or organism's body. So that particular organism have a special kind of a tissue, we call it as a placenta, which is a kind of a bridge between the mother and the fetus, and mother continuously provide the energy or nutrition to the growing fetus. But now what about the organism that usually gives or lay eggs? So when the eggs are laid down, there is no physical connection between the egg and the mother. So at such condition, the egg contain huge amount of yolk. Now coming on to this point, basically what we have said is that particularly the egg contains high amount of yolk, while some of the organisms egg contain little amount of yolk. Now this, on the basis of this, our cleavage pattern is also going to get differ. That means on the basis whether the egg is having large quantity of yolk or whether the egg is having a less quantity of yolk, we can have two different kind of a cleavage. One is basically a holoblastic cleavage, a complete cleavage rather we can say. Here you will find the egg or the yolk is, position of the yolk is isoleucithal. That means it is evenly distributed in the zygote. The yolk is evenly distributed. And such kind of a division, such kind of a yoke position suggests that it can show radial cleavage, it can show spiral cleavage. Another portion or another arrangement of the yolk is the mesolithal. That means here the egg can be, or the yolk is moderately present. It is not very less, it is not very high, but it is present in moderate amount. This kind of a division suggests cleavage pattern of bilateral, rotational. These are the two things that we are going to discuss. When a yolk is present in a larger quantity, we have a incomplete cleavage. That means meroblastic cleavage. Such cleavage are shown in such yolk arrangement. The position of the yolk is telolithial. That means it is located at one end. I'll give you an example of a chicken egg. You can find here the yolk is usually concentrated at one end, and one of the portion is remain empty. So such kind of a arrangement is called as a telolithial. Another is the centrolithial. This is usually found in the egg of a insect. So the yolk is arranged in the center, and it is such kind of a division shows discoidal or superficial kind of a cleavage. Okay, let's move ahead. The radial cleavage is shown, and the spiral cleavage can be seen. Also, bilateral cleavage and rotational cleavage can be seen. Let us come on to the radial cleavage, how it is going to happen. During the cleavage, the zygote will undergo a division where the cells are going to get divided in this particular fashion. This kind is called, this cutting or this age where the eggs get divided is referred as a meridional point. Just like a meridians on the globe, it is a meridional point where the cleavage furrow can develop and the two cells can be formed in this particular fashion. You can see in the top diagram here two cells are formed. So the division was like this. That's why the two cells are formed. Another one is the again a meridional cleavage. Again a meridional cleavage, it will result into formation of a four cell. One, two, three, and four cells. Now, after this four cell stage, there will be an equatorial. Equatorial means what? If you have this globe like thing, the equatorial division will be there. So here the equatorial division is there, and two tiers of cells are formed, four above and four below. Okay, two tiers of cells are formed. So usually when you look at the at the egg or the zygote which is undergoing a radial cleavage, you will find this kind of an arrangement, and in between you will find a cavity. This cavity is referred as a blastocele. It is referred as a blastocele. Who or which organism shows radial cleavage? Echinoderms and Amphioxus. Spiral cleavage is similarly the same. Here in spiral cleavage, the cleavage furrow is meridional. That means it will not top to bottom. It will be in the fashion like this. Or I'll just explain you. It will be like this. So the cells are going to be on the diagonal front. The cells are in the diagonal front, and the cleavage is in the spiral pattern, just like the balloons or just like the bubbles that are arranged in a soap bubbles. Bilateral cleavage here, once the egg is divided, the two dimensions or right and left portion of the organism is decided or determined. The left portion will always form the left part, and the right cell will always form the right part of that particular organism. In rotational cleavage, the cleavage pattern goes on changing. First, if the first cleavage is meridional, it will result into two cells, and these two cells will now undergo different cleavage pattern. One will be from this portion, that is the meridional, another will be the equatorial, and this will continue. In the meroblastic, that is incomplete cleavage, what we are going to see is that we are going to have these different the cleavage takes place only on the disc, and it is the complete cleavage does not take place. That means the egg is not divided completely. Because of the presence of yolk, the egg only divide partially, or half portion of the egg is divided. It is usually shown by the zebra fishes, and it is usually uh, in the organisms like reptiles and birds and fishes, or particularly in the pisces, it is this kind of an division is seen. In meroblastic cleavage, another pattern is the superficial. Here you can see the egg of an insect is there, and centrally located nucleus is there. The nucleus will start to divide number of time, but the cytoplasm is not dividing. So in a common cytoplasm or in the common yolk, you will find many nucleus is there. Later on, the nucleus will arrange themselves on the periphery of the egg, and then the cytoplasmic division or the cell cytoplasm will start to divide to form many numerous cell around this periphery. It will result into this kind of an structure. This is called as a superficial cleavage. What are the significance and importance of the cleavage? It converts a unicellular zygote into a multicellular embryo. This we have talked about. It also maintains the cell size and nucleocytoplasmic ratio of the species. Cleavage produces large number of members of cells or blastomere required for the building of offspring's body. And during the cleavage, quick mitotic division of blastomere occurs, following which there is no growth of the blastomere, and cleavage brings about the distribution of cytoplasm among the blastomeres. Reference book for this particularly I have referred Developmental Biology, third volume by Gilbert. Website also, you can see different websites you can also visit. Online ebooks are also available. Thank you very much.